Welcome, everyone. Today, we are diving into a foundational topic in evolutionary biology, the concept of biological races. If you have ever wondered how a single species can look so different across the globe, yet remain connected, you are in the right place. We will build our understanding from the ground up, exploring what races truly are, why they matter, and the mathematics scientists use to identify them. Let us get started. To begin, let us imagine a giant family of rabbits living in a massive, continuous forest. Over time, some of these rabbits migrate to snowy mountains, while others head to a hot, sandy desert. After thousands of years, the mountain rabbits adapt by growing thicker white fur to stay warm and hide in the snow. Meanwhile, the desert rabbits develop thin brown fur to blend into the sand and stay cool. Even though they now look completely different and live in entirely different environments, if you brought them back together, they could still have babies. This beautiful story of a single species stretching across the world and adapting to local neighborhoods is exactly what biologists mean when they talk about races or subspecies. Now that we have an intuitive picture, let us look at the formal definition. In evolutionary biology, a race is defined as a geographically distinct population of a single species that has accumulated genetic and physical differences. Why does this happen? Because each population faces unique environmental pressures, which leads to local adaptation through natural selection and genetic drift. But here is the crucial part. Individuals from different races of the same species are still biologically capable of interbreeding and producing fertile offspring if they meet. Therefore, a race is not a separate species. It is simply a distinct ecotype, or subspecies, living under the umbrella of a larger interconnected species. Building on that definition, we must understand where races fit into the grand timeline of evolution. Biologists view the formation of races as an early, intermediate step in the process of speciation. Speciation is the ultimate creation of entirely new species. When populations become geographically isolated, they start to diverge, forming races. If this geographic isolation continues for millions of years, the genetic differences become so vast that they may eventually lose the ability to interbreed altogether. Once that reproductive bridge is burned, they officially become separate species. So studying races is literally watching the very early stages of new species being born. Understanding this evolutionary timeline brings us to why this matters in the real world. Recognizing races is absolutely critical because it allows us to witness microevolution happening in real time, right before our eyes. In conservation biology, this concept is a powerful tool for protecting endangered animals. For example, if we know a specific race of tigers has unique genetic adaptations to survive in freezing climates, conservationists will work specifically to save that unique genetic lineage, not just tigers in general. Furthermore, studying these local genetic adaptations helps scientists understand how certain diseases might affect different populations of plants or animals. Without the concept of races, we would completely miss this fine-tuned diversity. To move beyond physical appearances, scientists need a way to objectively measure these differences. This brings us to a mathematical tool called Wright's Fixation Index, denoted as FST. Conceptually, this formula compares the genetic diversity within specific local populations to the overall genetic diversity of the entire species. Let us break down the symbols. FST is the fixation index itself, a number between 0 and 1. HT stands for the total expected heterozygosity, which is the overall genetic diversity if we combined everyone into one giant group. HS represents the average expected heterozygosity found within the separate individual subpopulations. If FST increases towards 1, the populations are highly isolated and distinct. If it decreases towards 0, they are freely mixing. Now that we have our formula, let us see it in action with a concrete example. A researcher is studying a species of beetle living in 2 different forests. We are given that the total genetic diversity of all beetles combined, HT, is 0.50. The average genetic diversity within each individual forest, HS, is 0.40. We need to calculate the fixation index to see if they are diverging. We substitute our values into the formula, subtracting 0.40 from 0.50 in the numerator. This gives us 0.10, representing the genetic variance strictly due to the separation of the forests. Dividing this by the total diversity of 0.50, we get a final value of 0.20. Physically, this significant number tells us the beetles are not mixing much and are becoming distinct races. Let us apply this same mathematical approach to a question closer to home: human populations. A geneticist wants to evaluate human population structures across different continents. We are given a high total diversity, HT, of 0.80 and a within population diversity, HS, of 0.76. We want to find the fixation index. We plug these numbers into our formula. Subtracting 0.76 from 0.80 gives a numerator of 0.04. We then divide this by the total diversity of 0.80. The calculation yields an extremely low final answer of 0.05. This means that only 5% of human genetic variation is due to geographic separation, showing that humans are highly interconnected. To give our human calculation some context, let us look at a species of chimpanzees separated by a large river. Here, the given total diversity, HT, is 0.60, and the within population diversity, HS, is 0.42. Again, we use Wright's formula to find the fixation index. We subtract 0.42 from 0.60, which gives us a numerator of 0.18. We then divide this difference by the total diversity of 0.60. This yields a final answer of 0.30. Physically, this tells us that 30% of their genetic variation is caused by the populations being separated by the river, which is acting as a massive barrier to reproduction. Now that we have calculated these values, let us prove what they mean biologically, starting with humans. We established in our example that the fixation index for humans is 0.05. Mathematically, this proves that a mere 5% of human genetic variation is due to geographic separation. The logical consequence is that the remaining 95% of all genetic variation exists within any given local population. Because the vast majority of diversity is shared locally rather than divided globally, we can deduce that human populations are genetically very similar across the entire globe. Therefore, we arrive at the biological proof that human populations do not form true biological races. We are simply too interconnected. Following the same logical derivation, let us look at the chimpanzees. We calculated their fixation index to be 0.30. This step proves that 30% of their genetic variation is strictly due to being separated by the river. In evolutionary biology, this represents a mathematically significant level of genetic isolation. The reasoning here is that the river acts as a strong, physical barrier that prevents the two groups from freely mixing and sharing genes. Because this isolation is so pronounced, we can definitively conclude that these chimpanzees represent true evolutionary races, or subspecies. Their genetic divergence is well underway. To visually reinforce what we have just proven mathematically, let us look at this diagram of geographic isolation. On the left, you see the original continuous population, represented by a circle with equal parts blue and yellow. Here, gene flow is high, and the fixation index is near 0. As we move to the right, a geographic barrier, in this case a mountain range, splits the population. Notice what happens to the genetics. Above the mountains, race A in the snowy environment becomes predominantly blue. Below, race B in the desert environment becomes predominantly yellow. The text between them highlights restricted gene flow and a high fixation index. This visual perfectly captures how environmental barriers drive the genetic divergence that creates biological races. We have covered a lot of ground today, so let us summarize our key takeaways. First, biological races are geographically separated populations of the same species that have adapted to their specific local environments. Second, remember that races represent a crucial intermediate stage in evolution, showing us how microevolution can eventually lead to entirely new species. Third, we do not just guess about these differences. We use mathematical tools like Wright's fixation index to objectively measure genetic isolation. A high index means distinct races, while a low index, like in humans, means a highly interconnected population. You now have a solid understanding of how life dynamically adapts across our diverse planet. Great job today.